Good afternoon, everyone, and welcome to today's ECETOC webinar on omics. My name is Megan D'Souza, and I'll be guiding you through this session. We're very pleased to see so many participants joining us for this important discussion. Today, we'll be exploring the growing role of omics technologies in regulatory science and risk assessment with perspectives from leading experts of the ECETOC Scientific Committee. A quick note before we begin, the session will be recorded and made available afterwards. We encourage you to use the Q&A function throughout the webinar. Our speakers will address your questions during the dedicated Q&A session at the end. With that, let's begin. So for our first speaker today, we have Dr. Tim Gant from Imperial College London. Tim will introduce the field of omics and walk us through ECETOC's history and involvement in this area. So over to you now, Tim.
Okay, hopefully that's showing okay. Does that look okay to you?
That's good. Great.
Okay, so welcome to all of you. It's my pleasure today really to walk us through really a very illustrious history of ECETOC in omics, and just to have a look back as to where we've come from and where we've got to as a way of setting the scene. And I thought I'd step right back, and my own involvement with genomics started in 1999 and 2000, and you'll probably never see one of these machines again, but this machine on the left here, shown here, is in fact, I was just looking for a pointer, I can't find one. Oh, here we go. This machine here is a Joe DeRisi microarrayer that we built in about 2000 in collaboration with the University of California, San Francisco, and also the NIHS at that time. And this produced microarrays using image clones, so cDNA clones, that were really very successful at the time, and so successful that even in the last couple of weeks, I've been analysing some data that we in fact generated in 2005 to find some more novel findings in respect to a different compound. And this array on the right here, shown here, is in fact one of our first ones. It was about 6,000 cDNA clones at the time, and it was just looking at drug resistance in cells. And this one really marked the start of my own particular journey with omics, and how I'll run you through how that came to pass in terms of involvement with ECETOC. So the ECETOC started in 2007, as far as I could go back, looking back through the involvement of omics and my own involvement with ECETOC doing omics. But actually there was involvement before that. But the involvement then was through the CEPHIC LRI program, and in particular through the External Scientific Advisory Board that I was then chairing for the CEPHIC LRI program. So there was involvement before then, a number of meetings, but the first ECETOC, actually ECETOC branded meeting I could find was this ECETOC technical meeting in Brussels, which took place in the Brussels airport in May 2007. And this is I've just picked some slides and some images from these meetings. And really by 2007, we were in fact remarkably exploring how much could be done with omics. And I was quite surprised at how much we were doing then. We of course started with microRNA transcript levels, and we're still looking at that. We'd already branched into epigenetic analysis, splice variants, translation analysis, which is particularly a niche area, never really found a role in regulation, but nevertheless was there. Transcription factor bindings, microRNAs, which of course found good roles in biomarkers in particular, and analysis of array CGH, and also at that time we were looking at transcription-based analyses. So these two are particularly mechanistic-based methods, not particularly finding a role in terms of regulatory application, but all of these others have in some way or other found a role in regulatory application. So that 2007 meeting in Brussels Airport really marked the start of omics involvement with ECETOC. As I said before, that was with the CEPHIC LRI program and the ICCA. And we then moved on to Malta in that same year. In fact, as far as I could tell, there were 3 meetings in 2007. And it's interesting how some of the issues that we were facing then, we're still facing now, and we still haven't found, in a way, ideal ways forward, and some of these still haven't been put into standard methods. But here, I just found from our report at that time, and then we were producing the workshop reports in the report format of ECETOC, that we were worried about exaggerated expectation. Well, we still are. Underestimation of the sheer complexity. Well, that still held us up. Difficulties in variable control and inappropriate experimental design. Well, how many of those messages still ring true today? And that was from 2007. We then move on to Malaga, and there were a number of meetings in Malaga, as you'll see in a minute. So Malaga seems to have been a favourite destination of ECETOC. And interesting there, I found from this meeting, which was in December in Malaga, looking at the application of omics technologies in toxicology and ecotoxicology, and particularly looking at case studies, that the interpretation of genomics was coming up then. And this is still something that we're working on now very actively, as to how you actually interpret genomics data and how you take it forward to actually get some real world outcomes and some applications in the regulatory aspects that we're interested in. And this here was a slide from that meeting. At that stage, we had primary data, primary statistics and filters, and then we're moving on to interpretation and then secondary statistics. I think probably now we'd invert these and we put interpretation at the end with secondary statistics here, being informed by then the metadata, the clinical chemistry, population pathology, etc. So we recognised that importance then. And then the purpose then of coming back to hypothesis generating and testing. I've only just seen, after all these years, I've just spotted the typo down there. So it's taken me, nearly 20 years to spot that typo. But there we go. So we've got interpretation now down here as an area that we're still working on. So moving forward then, we started to look at role and significance in detecting adducts and mutational ability. And this was a workshop in CAVTAT in 2008. I'm looking at the biological significance of DNA adducts, and this was in collaboration with the UKHSA program. And here we were looking at things like looking at the activation of microRNAs versus genes which were secondary in activation to something else happening in this formation. In this case, it was the formation of adducts on DNA, and then the repair of that DNA afterwards. And one case study we looked at here, which is one of many, but one that I presented, was looking at benzpyrene and the benzpyrene dialipoxide here in particular, and putting it into p53 null and p53 wild type cells, and looking at the different genes activated through the AH receptor pathway and the DNA damage receptor pathway. And we're starting then to get this interpretation, which with the metadata we could start to do, and was really starting to build on this interpretation principle that looking where there are more DNA adducts, you've got more activation of the DNA repair genes up here, with ATM activation down here, which are typical of DNA repair, with a 10-fold lower dose, so it's less activation of these genes. And then in the p53 null cells, you didn't get the ATM activation, so instead you started to see AH genes on a dose response basis. So really starting to really get a handle on how we can apply genomics in the regulatory chemical field, and that is work, of course, we're still continuing now. And then moved on to what I've called the further Malaga meetings. And there were 3 of these actually through 2010, 2013, 2016 that really started to really set a very firm foundation for our use of omics in toxicology and in ecotoxicology. And these 3 were exploring the omics and ecotoxicology case studies and risk assessment. This is workshop report 19. Omics and risk assessment science was in February 2013 and was workshop report 25. And then exploring the non-coding RNAs and risk assessment science was March 2016 and its workshop report 32. And these looked at some of the other areas. So we were looking particularly with the microRNAs at biomarkers, individual gene expression. The mode of action had started to appear here, subsequently, of course, developed into the adverse outcome pathway, and now developing into meetings that are coming up on looking at how to automatically transfer gene expression profiles to adverse outcome pathways. And that's a meeting coming up in Esprit JRC in October. And discerning adverse events and genetic modifiers and prediction of toxicity and adverse outcomes. What we're particularly interested in is, can you take patterns of gene expression at that stage in, for example, a 5-day in vivo test, and actually predict what will happen at a later time point. And some of that was really quite successful and is now being incorporated. And also we were starting to explore other areas here. So this is just one slide showing how we were looking at microRNAs. They were an emerging subject at that time in 2013. And this just happens to be microRNAs in sperm, where they're particularly relevant, that could be useful in, for example, developmental toxicity. And also looking at the exposure end here, looking at how epigenetic marks could be used as surrogate marks for historic exposure. So moving forward and actually starting to address that exposure element of the risk assessment profile by looking at the historical exposures through changes in methylation marks. And then, of course, there's hazard in there, and this was also put in Malaga, and really we were getting quite sophisticated then in terms of being able to look at both DNA methylation, messenger RNAs and microRNAs in the same systems to actually look to see how they were changing in relation to phenotypic anchoring, so pathological change, but also predicting that pathological change that was coming back through there, so use and prediction of hazard. So we're really kind of developing the technology and pushing it into application. So then that led to a very informative set of meetings that took place in Brussels in 2016. And these really evolved from the initiation of Alan Poole, who was then the CEO, shown here. You can see, I think, a very younger version of David, who'll come up next, and a few other people you might recognize on there, including a younger Rita Tong there as well. So we met in Brussels, and it was really to consider how to standardize the protocols for data analysis. And there was awareness at this time that depending on how you did the statistics, you could arrive at a different set of differential gene expression or differentially expressed genes. For example, it could greatly affect how you did the interpretation downstream. There were no OECD guidance documents, there were no principles established. So there was really a need to start to do this. And this small group got together to really address some of those issues. And what we looked at then was the work area from experiment to understanding, or what we might now call interpretation, which is put here. And what we were focusing really was this step here, going from data to gene list. And really this is only now just starting to be incorporated in the OECD guidance documents. And the reason we focused here was that we considered that data generation and the protocols for data generation had already been very adequately addressed by the MAQC consortium led from the FDA by Rita Tong. Metadata and data storage had been addressed very early on by Brasmer et al. In their Nature paper in 2001. So we didn't address that, and we didn't address interpretation because we viewed that as too complex, and there's work now ongoing within a task force at ECETOC to actually have a look at this, and I think that has underpinned that is a complex area in terms of the time that task force has taken and is taking. But what we decided to look at was this data processing. So really going from your raw experimental data here to your gene list, and how you do the data collection, the pre-normalization, the normalization, and a statistical selection of differential gene expressions to actually get you to that point where you can do an interpretation. And this was then incorporated in the project called the CEPIC LRI C4 project to actually do this work. And that was led by Florian Kimont at the University of Maastricht, and I'll come back to that. So then there was another key meeting in 2016, and this really took some of that work and put it into a wider context. And this took place in Madrid in 2016. And there was a special issue of regulatory toxicology and pharmacology that came out of that, with a number of papers in it that set forth both this idea of how to do this conversion of data from raw data to data that you're actually going to interpret, but also took it wider in going back on the work of Brasmer and looking to how to establish frameworks and put those within OECD guidance documents. So that work was taken ahead by a number of different people. So we had the what was called the transcriptome reporting framework then, and also frameworks for how to do some of the interpretation was starting to creep in here with a paper from Jim Bridges. So that was a really influential meeting and actually set forth the work streams that have led now to the papers that have come through from ECETOC and been published and accepted through ECETOC and the 38 member countries there. So this is how we went from Madrid to the OECD projects, and this is really the framework. And I'm not going to walk you through all of this. You can see this on the OECD website. But we've now got to this framework, which includes reporting and data analysis and reporting modules, ways for collecting and processing data from the various omics technologies. And you'll see we've got an expansion across here, not just in the nucleic acid level, but also at the protein level and the metabolite level down here as well. So they've all been incorporated within this framework. And really very recently, we've got now a guidance document on good practices and standardization of sample collection. And that's been led particularly by Mark Wyatt and John Coburn. So that's very recently been published. And this transferred in name from being the TRF, as it was named in Madrid, to the OECD Omics Reporting Framework, or the OORF. And the Cefic LRI C4 project was originally named as the ODAF here, which was the Reference Omics Analysis, or it was the Omics Data Analysis Framework. It's now been renamed to the Transcriptome Reference Omics Analysis Method. But this is the output from the C4 project led by Florian Quémont that is now being adopted into an appendix of the OORF, which was on the previous slide, and that's now in the commenting round of the OECD. So really, we've achieved that translation of knowledge from the 2015-16 meetings that really looked at this issue and brought it forward to a Cefic LRI C4 project that was then taken on by an academic partner and has now transferred that knowledge into the international framework under the OECD. So finally, in 2022, we went to, well, we weren't in Brussels, we were online, unfortunately, due to the influences of COVID. But this was to look at the ECETOC workshop on omics thresholds in non-adversity. And then there was a paper that arose from this. So really, we're transferring from technology-driven level to actually looking at how to use these technologies in understanding. And so this is really leading on now from that work into the interpretation work. So we go from Madrid to Paris, or I put from Madrid to Brussels and on to Paris. So this was really the work streams considered in 2016 in Brussels, led to these pieces of work in through Madrid and on through in Paris, and then the reporting frameworks here. And now the ongoing work that we're getting to in terms of the interpretation models, it's now a task force under ECETOC led by Florian and Richard Curry at Syngenta. So that's work ongoing at the present time. So we've moved from these processes of getting standardization in sample collection, getting standardization in the way that data is derived, data is processed, and now into the interpretation levels. And ECETOC has been fundamental in supporting that all the way through. And then we moved to the meeting organised by David, who's online, that was last year, AI and chemicals risk assessment, and really moving on now to look at how new processes in data processing and in language and image and data mining and modelling driven by the AI models can be used in genomics models and elsewhere, of course, but also on genomics data, not just to interpret genomics data, but to reuse and rebuild genomics data, even to derive in silico genomics data. And that's going to be a real work area as we move forward and how we use that in hazard and risk assessment. So in conclusion, from 2022, this is the pathway to regulatory adoption that we put up then. And you see it's really taken a long while now, nearly a quarter of a century to get to where we are now. are starting to really build confidence through these international protocols and publication through the OECD. And I think there has to be a big thanks given to ECETOC at this stage for sticking with the pursuit of this pathway for now more than a decade, in fact, nearly 2 decades. And I think we also have to acknowledge the support given to the hotels in Malaga. So I'll give you some thoughts from the early days just to think about. And this was from 2001, was John Quackenbush, who you might remember, published in Nature Reviews Genetics, that the methods that are used to analyze the data can have a profound influence on the interpretation of the results. Well, I think we've almost cracked this bit. We definitely haven't cracked that bit, so we've still got some way to go. This was from my old boss, Professor Lewis L. Smith, you might remember. New technologies impose greater demand on toxicologists to exercise expert judgment on the meaning of their data. That's as true today as it was then. And then finally, from Rusty George and Hydrin, gene expression might identify a hazard, but assessment of risk requires dose response data and mechanistic understanding by pathway analysis, particularly at low dose. Well, nothing's changed there. And that was from an ICCA LRI workshop meeting that took place in Amsterdam in 2008. So with that little trip through history and the very admirable involvement in ECETOC, for which we give you great thanks, I will just say thank you. And back to you, Megan, or maybe back to you, Ben.
Yep, so thank you very much, Tim, for that excellent overview and for setting the stage for today's discussion. So up next, our next presenter is Dr. Ben van Ravenzwaay of Wageningen University and Research. Ben will continue the story of ECETOC's work with omics, with particular insights into metabolomics. So over to you, Ben.
Okay, so I hope you can see the screen. It should be in presenter mode right now. Can you confirm?
Yes, we can.
Okay, that's good. So yes, the work that I will be reporting on is really based on metabolomics, and I'll try to give you some flavors of potential applications. It's work that's been going on for the last 20 years, particularly at BASF, but also at many other places. So just to place metabolomics briefly on the map, there's DNA, RNA, proteins, and metabolites, and the respective omics sciences or technologies on the right-hand side. Over time, we would come to the conclusion that metabolomics is actually pretty close to classical toxicology. And the reason why we started the Metabolome project at BASF was the hope, and it turned out to be a fruitful hope, that you might be able to catch within basically a single drop of blood changes, adverse effects in various organs. After all, virtually all of the organs that we have in an animal or a human being are in contact with blood. So the idea was, if there is like a liver change and form of kidney toxicity, we might be able to pick up those signals in the blood and actually with a single matrix determine multiple organ toxicities. So the technology is basically using a drop of blood, literally a drop of blood from basically any rat study, and then go through a metabolome analysis, which is basically LC, MS, or GCMS. You get a lot of signals, and out of those signals, we basically used about 300 what we call known metabolites. Over time, the platform has been expanded to various other, let's say, metabolites, be it hormones, be it other components of cells. But the idea is you take basically a sample and you compare that sample with your database that you build up over time, and you look for matches. Now, this is a nice video which will tell you get 100% match. Everybody who knows biology will know that you never get 100% match. But the idea is basically to compare a full fingerprint of your unknown substance with your reference database. And at the same time, within this larger fingerprint, look for smaller changes that are representative of specific modes of action or organ toxicity. And how that works, I'll try to show you in the next slides. So you have a test substance. It's basically in a database that at BASF was called MetaMap Tox. You look for a metabolite profile, and you compare that particular metabolite profile with all the profiles that over time you have been able to identify in terms of various modes of action. At the same time, you get obviously also a statistical correlation basically on ranking of biological similarity of the entire metabolome. And if these two match, and I'll give you an example later on, then you're pretty sure that you actually have identified a relevant mode of action. The basis for all of this, and that applies to any omics technology, is reproducibility, reliability, and by now also definitively transparency. And what you see here on the left-hand side of the screen are 700 controlled male and female rats. And it is important that these values, if you look at it from a principal component analysis, are actually relatively close, and that treated animals are spread out. We'll take a closer look at that and its consequences later on. But what you can also see that within this control group, you can see two lobes. And the resolution of that two lobes is probably obvious to many people, but males and females from a metabolomic perspectives are actually quite different. So mixing the two in any analysis is not a good idea. It also means that when you develop patterns of similarity or ranking of compounds, you always need to separate males from females. So here's an example. I've selected here within this database 3 compounds, cloverbrite, fenofibrite, and wyeth. They all have an influence on lipid metabolism. They're peroxisome proliferators. And what you see on the lower hand of the screen is their common metabolite change. So if you take in the center a cloverbrite, you see FH7. I hope you can see it. That means females, high dose, day 7. And the values that are displayed are changes relative to control. So if it's pink and you see 2.04, it means that the particular metabolite, in this case gamma-linolenic acid, has increased twofold in female high dose at day 7. And you can see the following days, day 14 and day 28, next to them. The yellow marks means it's statistically significantly down regulated. And again, the value is relative to controls. If it's not statistically significant, you can still see the relative change. So if you take these 12 metabolites and you compare these with the rest of the database, then you get an awful lot of other compounds, because peroxisome proliferation is something that's pretty common. You find fibrates, you find phenoxyherbicides. This list is actually truncated. You'll probably find 20 or 30 compounds by now. By the way, all of the work that I've been referring to has been published, and you can see the publications always in either the right-hand corner or the left-hand corner. So that's the pattern. As I indicated, you can also do a full comparison of the entire metabolome, and then see, okay, what's the ranking of all the other compounds in the database? And here I've chosen cloverbrite, again, peroxisome proliferator. And if we take the Pearson correlation, which is, I guess, the most common one, and you see the ranking, then next to cloverbrite, the highest rank is fenofibrate, mecoprop, and well, the other 12 compounds that you've got here are all peroxisome proliferators. So in this case, you have really the combination of an identification of a mode of action as well as a ranking of your compounds that you're interested in relative to others that indicate, yes, there's a very, very strong connection to a particular mode of action, in this case, peroxisome proliferation. Here's another example, ETU and PTU, 2 compounds that inhibit an enzyme called NIS that is important, essential, in the formation of thyroid hormone formation. And with inhibition of this enzyme, thyroid hormone formation is reduced, and that has obvious consequences. And in terms of changes in metabolites in the blood, you can see them over here. It's a pretty long list. If you use this pattern and you go back into the database, you'll find methimazole, which is an analgesic drug that's more, I think, over the counter in Europe, but there's still a lot of places where you can get it. It has the same pattern. And you see metiram, which is a pretty old fungicide, which has the same effect on the right-hand side. Now, the metiram example I selected here to indicate that you can recognize such a pattern only based on a 7-day study. So you only see MH7, male high dose 7. And even with that, you can identify metiram as a compound that's a direct inhibitor of thyroid hormone synthesis. The application of that is actually pretty clear, and that was always one of the targets that we had at BASF. Basically, fail early, fails cheap. You would identify in your earliest study of multiple applications, and that doesn't, or it's usually, let's put it like this, a dose range finding study of 7 or 14 days. So you take a drop of blood, you compare it with the rest of the compounds in the database based on your metabolome research, and you're able to identify, in this case, a compound that has a direct effect on thyroid hormone formation. And based on that, you're pretty sure you can expect thyroid tumors, maybe some problems in developmental neurotoxicology, developmental toxicity. It's an endocrine disruptor. So given the current context of regulatory activities, you're probably well advised not to continue with the development of such a compound. So that's basically prioritization and early detection, helping you to make hopefully wise decisions in compound development. So just as a summary, I would call this reduction through refinement. You're actually, identifying modes of action at a very early stage. You can focus on those things that are important if need be, or stop development if you think that the modes of action that you found are not appropriate. At BASF, when I left BASF more than 3 years ago, we had around about 120 modes of action patterns established. The database contained more than 1,000 substances. You can identify molecular modes of action. And we had a prediction rate relative to pathology that was, I think, at that time at 84%. And also of importance, if you're interested in sort of risk assessment, we observed that the sensitivity of metabolomics is actually pretty similar to classical toxicology. So this is where I'd like to basically continue with other applications. This is really at the stage of identifying modes of action and making compound selections, if you wish, the development of green chemistry through a new technology. The most successful tool to reduce animal testing is read-across. Read-across is far from simple, and the requirements based on ECHA's RAF are actually pretty high. And I thought it was interesting to see if metabolomics as a tool could provide, let's say, a biological approach or an enhancement of structurally based read-across. And so we constructed this case where we have 3 phenoxy herbicides, where micoprop would be the target compound and MCPN24DP the source compound. And it's done in such a way that structures are similar, acute toxicity, kinetics is all similar. So this fits into, let's say, a classical read-across case that would be digestible according to the RAF. And we then did, or used metabolome analysis. Okay, we got these 3 compounds that from a structure point of view and a couple of other things are actually similar. How's the similarity when you look at biology, in this case, metabolomics? Well, you can see that here we now have like 25, 28 different metabolites that are all changed in the same way. So that indicates, yes, it is actually quite same. When we look at specific modes of action, you can see that it's liver, food consumption, some effects on the kidney. They're also very similar. And what I didn't show is all the negatives, but obviously when you've got like 5 or 6 positive, you are talking about 110 negatives. So the profile overlap of the 3 compounds is actually very, very similar. And the target organs are liver and kidneys. Now, the next step would be, okay, you've got these 2. Which is actually the better one? So here we now compared the 3 compounds, and looking at which metabolites are specifically changed. So you can see that if MCPP is your target, then 2,4-DP has actually more specific changes in metabolites that are comparable with MCPA that are comparable with MCPP than MCPA. The list on the top is pretty much longer than the list lower. And if you look at a PCA analysis, it's quite nice to see that there's a virtually complete overlap between 2,4-DP and MCPP. So the conclusion was that actually 2,4-DP is the best read-across source substance in this case. There's far more to this. There's also a quantitative approach for which I don't have time to explain, but it demonstrated this basically as a case study that metabolomics can enhance or can help to come up with a biological approach to read across. Now, where there's similarity, there may also be dissimilarity, and I've got two compounds that are pretty similar, 2-AAF and 4-AAF. From their biology, however, they're actually quite different, and with a Tanimoto score of 63%, they are structurally actually quite related. When you take a look at the metabolome of the two compounds, so on the left side 2-AAF, and the middle it's 4-AAF, you can see that they are actually quite different. And when you look at the compound ranking, then 2-AF has a very, very low match compared to 4-AF. It's on rank 1,443. So here's a case where two compounds that are structurally actually quite similar, but biologically very different, one is a liver carcinogen, the other is not, are also from a metabolome point of view quite different. The next one is, I guess, the tricky one. Here we've got 4 compounds. They're so-called HPPD inhibitors. They're a pretty new class of herbicides. And out of these 4, only the coumarin 1 and 2 are, according to the similarity, the Tanimoto score, similar. The other 2, nitrazone and topramazone, are structurally very different. When you look at a metabolome comparison, then you see that all of these 4 are actually very, very similar. Yes, the 2 coumarins have actually one additional metabolite in common, whereas the other 2 don't. And if you look at, on the left-hand side, the comparison against all other compounds in the database, and again, we're talking about a few thousand of comparisons that you would see, all of these 4 compounds rank within numbers 1 to 10, depending on low dose, high dose. There's no other. So here we've got compounds that are structurally very different, but from a biological approach, actually very, very similar. And theoretically, a read-across case based on biology should be possible here if the work is done in an appropriate way. This now is a very, very interesting potential high impact application, and that is related to the use of metabolomics for UVCBs. UVCBs are compounds with basically unknown structure. So if there's not a known structure, then it's basically impossible to base your read across on structures because they're not there. So the question here would be, or was, is chemical grouping and read across possible based on the metabolomics approach? Now, what you see here from L1 to L12 and BASF1 to 3, these are streams from a stream tracker. And simply by looking at, take L1, you can see that there's a number of marker substances. But if you add these all up, you don't even get to 15%, which means that 85% of L1 is unknown. And you can go through L2 up to BASF3. You hardly ever get above 50 to 60% of known compounds, and the rest is basically an unknown mixture. So how would you deal with that? So what we did was to run for all these studies, for all of these compounds, metabolome studies, and to see, can we actually base chemical grouping on the biological outcome. And of particular interest, I will get to that, are BASF2 and 3, because they're actually, this is the same Cas number, so the same mixture, but produced at two different sites. One is the steam cracker in Ludwigshafen, the other one is the steam cracker in Belgium, in Antwerp. And the answer to that question is an in vivo metabolomics-based approach for chemical grouping possible is yes. There are various forms how you can group them in terms of statistical approaches. Here is an hierarchical clustering, which is probably the most easiest one to grasp. So on the top, you see the controls coming together. And if we focus now on the very low end of the picture, you can see BASF 3, 2, and 1 clustering together, and actually the lowest distance between BASF 2 and 3 is actually found for these or this Cas number, this mixture that is indeed the same. So that's it was a very pleasant outcome. We weren't sure what we were going to get it, but it demonstrates that this technology actually is able to group compounds, and that it provides a biological-based approach for chemical grouping of UVCBs. And as I said, there is no other option for UVCBs. Either you do for these compounds all the studies necessary, or you think about a biologically-based approach, in this case metabolomics, but I think that other omics sciences would also be suitable. Now, of the work that I've been talking about so far was done at BASF, and I think we got quite a bit of traction and interest. But the question obviously was, can such a technology also be transferred to other labs? So LRI helped us to sponsor an important project called Matching, where the samples from 8 different substances that were selected by ECHA and BASF were sent to 6 different labs in a blinded way, and they performed their metabolome analysis on these blood samples. So the blood samples came from these 8 studies that were all the same. And the question was, can they group? So the 8 compounds that were selected would fall into 3 groups, 2 groups of 3 and 1 group of 2. And the question was, do you get it right or not? And this here now shows the importance of data quality. So on the right-hand side, you see 6 plots and a red dot. The red dot are the control samples from a technical point of view, and they should cluster together relative to the blue axis, which are all the other compounds. And as you can see here, in 5 out of 6 cases, the red dot is far smaller than the circle that takes all samples into account, which is logical because you got an effect in these other samples. One exception, and that is number 6, where there's a huge overlap, which means that basically there's huge variation in the controls already. And as such, it is probably not a surprise that 5 out of the 6 labs got it right, and the 6th one wasn't able to group based on the technical procedure. The grouping itself is shown here. Again, 2 examples. One, hierarchical clustering at number B, and display number E is a PCA. And you can actually see that, yes, the groups are formed in all cases correctly, which is basic confirmation that the technology used in each group had its own type of technology, did not really matter as long as you got it right from a technical perspective. And the work that Tim has just mentioned about the sampling procedure that is now an OECD document is one of these examples how important a consistent manner of sampling is. I didn't only want to stick with in vivo work. There's also quite a bit of work that we've done on metabolomics in vitro. It was, I have to say, unfortunately, a very, very long journey. It took us 4 years to get an in vitro system actually working, which was far longer than I ever expected. And even though it worked quite well, it was so expensive that actually nobody was very much interested beyond, let's say, some special cases related to science. So a couple of years later, I had a PhD student of mine, Sabina Ramirez, working on the miniaturization of this in vitro product, and that worked, fortunately, and it demonstrated that the transition from in vivo to in vitro is not only pure technology, it is also related to resources, and in this case, basically the amount of money you have to invest to run such a study. But I'm very happy to say that, yes, it's possible. And because we're now working in 96 well plate, it's possible to do a lot of investigations. And what you see here is a plot that takes into account dose and time responses, in this case for a compound called nitrofurantoin, because the time response is often neglected in vitro studies. So the positive control that we've used is benzofibrate. You can see that on the left side, and you can see that starting from the controls at 3 hours, there's still a response that's close to controls, and then 6 hours, 24 hours, and 48 hours, the response, the metabolome response that we see is getting stronger and stronger. And the same applies to nitrofurantoin. There's a dose and time response, and we looked at the effects up to 48 hours. From that, you can actually determine a point of departure, which is essential if you want to do risk assessment based on in vitro studies. But then, of course, the question becomes, okay, now we've got this time response. How far should we take it out? So we looked at the point of departure, 3 hours, 6 hours, 24 hours, and 48 hours. And it turns out that although the response at 48 hours is stronger as such, the point of departure actually did not change. So we're convinced that with this type of technology, a 48-hour exposure time point is sufficient. But it was a wake-up call. You can't go to earlier or to earlier time points because then you would lose sensitivity. So a brief summary of the applications of metabolomics. In vivo, identification of mode of action and ranking of compounds, that's helpful for selection of compounds and understanding toxicity. Improved quality of read-across cases, read-across for UVCBs. I did not talk about the use of omics technologies in smart studies, but I think that has huge potential to do better in vivo work, and as such, reduce the amount of testing through refinement of the study. And I also believe that metabolomics will be essential to understand the contribution of the microbiome in toxicology. Further, as I indicated, metabolomics in vitro, early identification mode of action, and determination of point of departure for next generation risk assessment. With that, I'd like to thank you for your attention. And the picture that I show here is the Corinth channel. Apparently first attempted to be constructed in the first century of our time counting. It failed. Apparently, Emperor Nero himself tried to do it. It needed an important and explosive innovation, and after that, the channel was completed within 2 years. Sorry, within 12 years, not that fast, but it does indicate to me, don't give up on a good thing. It just needs innovation, and then basically continuing with that work and convincing others. And I hope some of you at least are convinced that there are applications in regulatory toxicology for omics. A big thanks to ECETOC, to all of my former colleagues at BASF, the LOA Consortium, and ECHA in Helsinki. Thank you very much.
Thank you, Ben, for those insightful reflections on metabolomics and its role in advancing the field. Our final presentation today will be given by Dr. David Rouquié from Bayer. David will be sharing his perspective on the future of omics, including the exciting potential role of AI. So over to you, David. David, I think you're muted.
Yeah, good afternoon, good morning, everyone. Yeah, I'm just sharing my screen. Is that okay?
Yes, it's perfect.
Okay. So let me just put my camera on also. Oops, sorry. Is that fine now?
We still can't see you, David.
Okay, so let me see, because I'm on my, must be better like that. Yeah, now?
Now we are unable to see the slides. We see
Okay,
you,
still?
and not the
You
slides.
see? Ah, okay. Sorry for that. Here we should go. No?
Yep.
Okay, good. Yeah, sorry for the glitch from at the beginning. Yeah. So yeah, it's a pleasure for me to provide you with a short outlook for the use of omics in regulatory applications, and I will be touching upon few examples about the use of AI in that context as well. So during my presentation, I like to start with yeah, some considerations related to the use of omics in the context, mostly on the context of next generation risk assessment, so NGRA. I will provide and try to express some few overarching challenges we may face. Then provide some opportunities about the use in particular of AI, and then finish by some things that already Tim mentioned, but some of the activities we are doing at ECETOC in that field. So talking about next generation risk assessment, this representation, this slide in that case represent the spectrum of different evaluations that can be performed for chemical risk assessment, and you have the full spectrum going on the left, the right-hand side with the pharmaceutical product, where basically all the batteries of evaluation are performed, from in silico, in vitro, lab animal testing during the preclinical phase, and human testing in the clinical phases. And on the other side of the spectrum, you could see the cosmetic products, where mostly, at least in Europe, in vitro and in silico and in vitro methods are used. And one of the questions at the present time, when it comes to reduction of animal testing, is to see where to position the evaluation of the agrochemical product, but not only that, the industrial chemical ones, and to set the bar at the right place. Obviously, we won't go into the side of the pharmaceutical product, but to which extent we should go toward the cosmetic products. And to make a long story short, I think the converging view now at the present time is to move toward a combination of in silico, in vitro, and short-term in vivo studies enriched with some omic endpoints. That has been named with different names. So at ECETOC, we call them SMART studies. In the US, they might be called also ETAP studies. In some other project in Europe, in the project PARC, it's called TG Plus, like Test Guideline Plus. And that's essentially this, in that context, that we have developed this representation, and that we have recently published a short paper in toxicological sciences, where in a joint session at the last SOT with ECETOC and SC, we developed this representation. And so during my talk, I will be referring to the use of omics data, not only into the in vivo short-term mechanistic studies, but also in the context of in vitro-based methods. So talking about omics in general, so one can consider this technology as high content biological assays, providing several big advantages. Being able to generate rich multi-parametric data on cellular and molecular responses for a wide range of cell culture. It could be 2D or 3D cultures, and of course tissues when it comes to short-term in vivo studies. They enable chemical grouping based on bioactivity pattern, and Ben showed us one application using metabolomics, but that can be done by different other technologies as well. They have been used to support the identification of benchmark concentration for biological activity, so the determinations of the so-called POD, point of departure, both in vitro and in vivo. And also they provide insight into a mechanistic understanding about how a molecule can induce some adverse effects. So to try to put a little bit in a larger perspective the use of these technologies, I tried to sum up a little bit how we characterize the adverse outcome induced by the chemical exposure, globally speaking, as a first start from in the way to chemical risk assessment. Traditionally, we've been using in vivo testing, and it's from the phenotypical evaluation of target tissues that the adverse outcome have been characterized first in vivo in lab animals, and that was used then to extrapolate the safe dose of use in human situation, human exposure. But obviously, before observing this adverse outcome in lab animal testing, some mechanistic and molecular events are taking place. So basically, the full genetic flux of information is at play. So the expression at the modification at the level of messenger RNA, translation of proteins, metabolites, and of course, that's also a big advantage of this technology, being able to evaluate and even before the time where the phenotype is observed in a target tissue. Nowadays, with this NAMs, on new approach methodologies in the context of next generation risk assessment, in vitro test methods are also being used using cell culture. The same phenomenon, the same steps are there, starting from the expression level at the protein, at the RNA, protein, or metabolite. And all the big questions about this new way of evaluating the effects of this molecule, the molecule, is to establish somehow the correspondence between those effects which are observed typically in vitro with the one which would be observed in vivo, could be in lab animal or in human ultimately. And among these different chain of molecular changes, there will be also a need to evaluate the interdependency, or how much characterize more the effects induced at these different layers of the effects induced by the molecule, both in vitro and in vivo. So what I try to summarize on that slide, naming it the big challenge, in the sense that those various omics technologies describe specific layers of the universe of chemical induced effects, globally speaking. And there is a strong need to really better characterize for each layer in their corresponding test system different things, in particular the natural variability of the measurements of the individual descriptors. I would call that descriptors. So it could be the transcript, the set of transcript, the proteins, the metabolites, or the morphological profile when it comes to phenotypic evaluation in cell-based assay. There is also a need to better understand the dynamics of all those measurements, omics measurements, both in vitro and in vivo. Dynamics in terms of dose, dynamics in terms of time. Ben mentioned that also a little bit earlier. To some extent, the reversibility of the effects. also should be investigated, and also the dynamic range of the different responses in order to better get informed about the significance of all those changes. And out of it, there is still the question. There are some evidence, but there are some strong need also to better understand the proportion of the biological knowledge that they provide with regard to the adverse outcome in general. So all this to say that the big challenge in all this is not to get lost in all those translations to make a proper risk assessment of chemicals. So I mentioned a couple of challenges, and I would like to illustrate two of them quickly. One related to the physiological relevance aspects. This is part of a work we have been doing a few years ago, where in a large data set of something like 8,000 chemicals profiled in several cell lines using transcriptomics, we were able to just describe the biological responses induced by all those 8,000 molecules in two different cell lines, VCAP and PC3, which are highlighted there in red and blue, each dot representing one chemical in a given cell line. So it's a two-dimensional plot called a t-SNE plot. And the distance between the plot inform about the degree of similarity of the transcriptomic profile. And so what you can see out of this t-SNE plot is that even if you're talking about two cell lines derived from the same tissue, so in that case the prostate, you will see barely few overlaps in terms of biological responses induced by these molecules, these 8,000 molecules in these two cell lines. Questioning a little bit how, I mean, the selection of the which cell line to be selected for an evaluation in vitro on prostate in general, if you want to evaluate potential effect of chemicals on the tissue, on the prostate from human being. The same was observed in lung cancer cell line, two cell lines derived from the lung, and there are also some very few overlap between the two cell lines in those results. Time translation challenge. This is also, as Ben says, something probably a little bit overlooked still. And this is an example I like to provide you with when it comes of a work we recently done in the frame of a PhD thesis from Vanille Lejal. There it's the data set of 110 chemicals in 3 cell lines, 3 time points, and 2 readouts, cell painting and transcriptomics. Same types of representation. It's not t-SNE in that case, it's UMAP. But in that case, each dot, again, the same principle, each dot represents one molecule in a given cell line at the given time point. And the distance between the plot inform about the degree of similarity of the responses. And as first sight, you see that according to the time, the biological responses look very different. And they are different within the different cell lines, but also across the different cell lines. And when you look at the UMAP on the left-hand side, on the right-hand side, sorry, on the L1000, it's a transcriptomics, so it's a gene expression technology. There also you see a very different set of pattern of the effects induced by the chemicals, which tend to be a little bit more conserved in transcriptomics compared to the cell painting, but still very much different. So this was just 2 examples of challenges in the field of the use of omic technology in the context of in vitro-based methods. I like to end my talk by providing you with a couple of opportunities. One opportunity lying into the predictive, what I would call predictive AI, and in that case, it's about prediction of molecular initiating event. So in that work, what we have been doing was to evaluate and compare the prediction based on compound structure when it comes to the potential of a compound structure to interact with a molecular initiating event, either predicting from the compound structure or from the bioactivity derived from a transcriptomic profile from cell culture. So on the left-hand side, you have the classical QSAR approach, where you have a training set with a series of examples of compound structure with a given property on a given target. You train a model to learn the relationship between the structure and the outcome of and the properties of the molecule. And then you can make use of this model to predict the properties of a new molecule based on the knowledge you have learned from your training set. Signature models are also possible. So in that case, you train your model not from chemical structure properties, but from gene expression patterns, and then you associate the same types of properties toward this bioactivity pattern to train the model and do the same thing, except that the input to interrogate your model is the new signature of a new molecule, and then you ask the model to predict if the molecule is going to be active, and in that case on the target that you have trained your model for. So just an outlook about some of the results we obtained. So overall, we demonstrated that using transcriptomic profile, signature model were possible, and sometimes almost outcompeting compared to the QSAR models. Interesting thing is that the models and the performances of the models really depend on the test system, so the cell line which has been used, because depending on the set of genes which are expressed in the cell line, that might have an impact on the prediction. But overall, we've done this exercise on 69 targets, and for 25% of the targeted prediction tasks, we saw that signature performed similarly or sometimes even better compared to the classical QSAR models. First opportunity. Second opportunity I like to highlight is about generative AI. So then I refer back to this predictive model using QSAR models. And in that case, that's something we've done in the lab with in collaboration with some colleague in pharma at Bayer. So in that case, the thing was to train a model to learn the relationship between chemical structure and the bioactivity induced by this chemical structure in a given cell line. But there are thousands of examples, right? Then we train a model, and basically what we've done was to put upside down the QSAR model by, instead of inputting a new molecule, we input a specific bioactivity pattern, and we ask the model to design a molecule which will have a high probability to induce this bioactivity pattern. So it's not per se something that's going to have an impact directly on the chemical risk assessment in the near future. It's more an outlook about what could be the future about chemical safety by design. So optimizing the chemical from start to get a safety profile which is optimized from the very beginning. So we did this work and published those results, and more recently we made some experimental evaluation of those molecule which had been de novo designed by the molecule, and we can show that those molecule have nice properties in terms of bioactivity in some cell assays, and that a decent amount of them are doing the bioactivity that was designed for in a nutshell. So I like to quickly mention a new technology that I will categorize also in the field of the omics, which is cell painting, which is gaining some traction since a couple of years, or a little bit more, 3-4 years. So this technology called cell painting is a technology which was developed by Dr. Anne Carpenter at the Broad Institute. It's an in vitro high throughput method where the overall principle is to evaluate the bioactivity induced by chemicals, looking at the phenotypes of the cells cultivated in microtiter plates. So the principle is to grow cells, you expose your cells. After a precise period of time, you fix the cells and then you paint them by different fluorescent dyes, which allows you to extract some characteristic of those images, which are related to the pixel itself. So you do some what we call segmentations of the images in order to characterize the distribution of the different pixels from the images and define some morphological profiles. So this was initially the way to perform this morphological profile. Now we have also more fancy approaches using deep learning methodologies to determine and to characterize this morphological profile. But in a nutshell, those morphological profiles have been shown to be sharing similar properties if molecules are inducing similar effects. So it's a way to categorize the molecules according to different bioactivity, and from there infer about for example, some properties related to acute toxicity, mitochondrial toxicity, developmental tox, or liver tox, or also defining some molecular initiating events. This is brand new in the field, but it's gaining a lot of fractions, as I said, over the last few years. And I'd like to end my presentation to highlight two task forces we are busy with within ECETOC related to what I just presented you previously. One is about the definition of and proposal of a design and the use of smart studies, so short-term mechanistic studies in the context of NGRA. And also another task force, more recent, related to the omics data interpretation framework, mostly going toward the use of these omics technologies in the context of European regulation, where classification labeling of the effects induced by the molecule are needed when it comes to registration of the molecule. And with this, I'll stop there. I thank you for your attention and into the Q&A.
Many thanks, David, for that forward-looking perspective and for highlighting where omics research might take us next. We now have a bit of time for a quick Q&A session. So please continue to submit your questions in the chat box or raise your hand directly to pose your questions to our speakers. I see that we have a few questions in the chat box. So starting with the most recent question, we have here a question to David that states, would you expect a similar outcome related to time if NGS was used instead of L1000?
Would I expect different? Sorry, I didn't get the question. So expect the similarity between the gene expression according to the time. That's right. But I didn't get the in comparison to what?
Yes,
Sorry.
David, that's correct. You got it actually, because L1000 is kind of limited in terms of genes that you monitor. So that's
okay. Yeah,
right.
I see. You're talking about more the coverage about the biological
Right.
information, right?
Yes.
Okay.
Yes, that's correct.
Yeah. Alors, that's a good question, and honestly, I cannot answer it because I don't know. But maybe I can speculate a little and at least try. From what we what I would anticipate is that you cannot. Yeah, I could see that you would have the potential to see some gene not represented into the L1000 having different kind of behaviors in terms of time. Due to the fact that the selection of the 1000 genes which have been done at start are really the one which are really representative of a large series of biological information. But you cannot exclude that you will have outside this space different other genes which will behave differently, I would say. But the short answer, sorry, I must say, I don't know.
Thanks, David. I see we have a question for Ben. The question goes, I am delighted to see that in vitro metabolomics can be applied. You have emphasized the importance of dose and exposure time, but what about the specificity of the response depending on the cell type and culture method?
Yeah, that's a very good question, and the answer is at this stage, it would still be, let's say, cell type according to a specific organ by organ. You cannot expect that a liver cell culture will give a definitive answer on kidney toxicity. So there are indications that you can see differences, but I would not speculate on it. So yes, if you want to have a full overview of potential toxicities or organ toxicities, you would need, let's say, cell cultures of the major organs you are interested in. Obviously, the liver is in toxicology #1. That's why we started it, and most modes of action are pretty well known. But yes, the answer is it's not only time, it's not only concentration, but it also depends on the cell type.
And the question also continues, if you had to choose only one cell type, would you choose HepG2 today?
I think the best choice that you can make is a balance between a cell type that is, let's say, closest as possible to the human situation. That's the side that you really want. The other side of the equation is stability. So if you have, let's say, freshly harvested hepatocytes from the organ, then you have very relevant cells as such. However, their fluctuation is pretty large. They come from I would call a cozy in vivo situation. Now they're brought into cell culture. That is pretty tough. So these cells are trying to adapt to a completely new situation, and as such, although the cells are more relevant in terms of their competence, you introduce a tremendous amount of noise, and it's the balance between a very stable, let's say, tumor cell line versus rather unstable, freshly harvested cells. So it's you have to try and find it out. It was easiest to start to work with a liver carcinogen cell line. Given the situation that we have the technology now that's relatively cheap, I would definitely try other cell lines as well and see how far you can take it with, let's say, the most relevant one.
Yeah, maybe to add on what you just said, Ben, because it was something which was one of my highlights as a presentation. I was attending the EuroTalks last week, and there was a very interesting presentation by Professor Laura Suter-Dick in Switzerland, describing all the different aspects in cell culture, cell-based methods, to really monitor closely, starting from the cell media culture. The selection of the cell media culture has a huge impact from start. Of course, the test system, the condition of use, the technology, and so, yeah, the physiological relevance. This is really important aspect, yeah, to consider and really know the details.
So one more question, which I think is once again to Ben, that goes, are we building our databases on rat omics data, or are we already using human omics, from example, the pharma industry?
Yeah, so the data that I am familiar with are rat data. It will be definitely of interest to see how the connect between rats and humans will be. I'm not very optimistic that this was going to be easy in an in vivo situation. However, the opportunities are now fully there to take a comparison between the cell types in an in vitro situation. And if you think about the classical rectangle, human in vivo, rat in vivo, human in vitro, rat in vitro, we now have the option with the omics technologies to ask far deeper going questions and get multiple answers. So yes, I think the comparison on omics level in vitro is currently being investigated and is occurring, and it will inform also about let's say mechanistic differences between rats and humans.
So one last question to all our speakers. If one of the ultimate goals is to replace animal testing with omics-based technologies, what are the most significant technical hurdles or challenges we need to overcome to get there?
I'll give it a try. Actually, I would not start with a technical question, but rather with a philosophical question. You can have perfect technology if you do not have a framework how to use such a technology for regulatory use. It's, I'm not going to say it's all in vain, but you're not going to see much progress. So it does need, I think, in addition to technology, a cultural change. And I'll leave the technology questions then up to Tim and Dave, but this is very important to understand. It's not only about technology, it's also about trust and really accepting a change.
Yeah.
Trust comes into that, Ben, and I think actually in the challenge in terms of replacing animal testing with sort of, you're not replacing it with omics-based technologies because the technology is applicable to in vivo or in vitro, but using omics-based technologies in conjunction with an in vitro system to replace an animal system is that, of course, the omics technologies give you a much wider landscape of what's happening in those cells. So you've got much more to look at. So it comes back to an earlier question that I answered in the chat, which is to do with perturbations in gene expression can be homeostatic, adverse, or adaptive. And of course, you're going to see all of those in the in vitro system, but they might not be necessarily modeled in the in vivo system. So you need to understand what's relevant and make sure the relevant ones are modeled in the in vitro system to come to the point where you can use omics technologies in conjunction with that system as a replacement for the animal system. So basically to use it to validate that system. So because it does give you that wider perspective, it does make that more of a challenge, but it also provides more opportunities.
Mm. Well, I'm gonna do a try to make it a little bit technological, but also philosophical all at once. So if personally, and that's my current state at the moment. So in my thought, I think one of the super big challenges we're facing when it comes to the implementation of NGRA in the larger sense, in a nutshell, is related to the fact that biology is noisy, in the sense that we have tons of confounding factors, which are inherent to what we are doing in vitro in particular. And It's not to say that, and we know that in vivo evaluation also, there are some things we can discuss about the reproducibility or in vivo testing in general, but the nature of the evaluation performed in vitro and the amount of data that can be generated, and having this perspective of the use of AI in a short term period of time, I think that for me the reducing and monitoring the confounding factors related to biological variability is something where I think we need to really pay attention to.
Okay, so with that, looks like we are at time. So that brings us to the end of today's webinar. On behalf of ECETOC, I'd like to thank our speakers, Tim, Ben, and David, for sharing their expertise and perspectives, and of course, to all of you for your active participation. We hope that this session has provided valuable insights into the topic of omics, and we look forward to continuing this important dialogue with you. So thank you once again, and have a great rest of your day.